So let's go to the next aspect, that is COVID. So even in your syllabus in coming years, this COVID would be updated in CBSE also, if I'm not wrong, because it's one of the pandemic, just like cholera at that one point of time. Now, how you study about mosquitoes, this mosquito dengue, mosquitoes as a vector, malaria, dengue, like that. Even corona also you will be studying in future. So let's see how the infection actually progresses in the body and system, and how it is actually causing differences in the body. Okay, we are talking about a normal COVID-19 virus. Okay, so let's move forward. Right. So you need to understand one thing, that the virus cannot be living if it is outside the living host. So it is always kind of an obligatory. Until it finds a living host, it cannot be living. It means it cannot replicate in the air, non-living systems. So even when it is coming from droplet infection, okay, when someone sneezes and touches nose and eyes and then touches the other person, the fluids through this body fluids, it is entering. It means that until it stays in there, that's why we are actually having the safe distance of almost 6 feet. We are maintaining. Why? Because the droplets would be heavy. When someone sneezes or talks, the droplets which comes out of the mouth and nose will be having a trajectory. It means in a parabolic path, because of the weight, because of the density, they fall in this curve. So that when we maintain a safe distance, obviously they will not be falling on us, and then there won't be any infection. We may not inhale also. So in that way, a virus needs a living host for its replication. So I told, right, any living organism actually tries to reproduce. So that's one of the living character. But when it comes to virus, we say it has replication, and when it got a suitable host, only it will be dividing. Okay, it's not this coronavirus you haven't seen in any plants. It means it is specific, species specific, right? So let's see. So the virus, corona means it has a crown-like protein structures around it, and then inside you have a viral genetic material. I told you, the genetic material can be of DNA or RNA, single stranded, one copy, or single two copies. It depends from virus to virus. It varies. Okay, so let's see further. So here each layer has its own speciality and function. So outer envelope spike proteins which interact with our cells and they attach, and then they will be entering into our human cells. And there would be a protein code which actually safeguards the internal genetic material from desiccation, because even viruses which has to travel from person to person within time span, if they have to move, so that it will be saved, the internal genetic material will be saved by this protein code. So here it's like this spike proteins are like a key, and our cells will contains lock. So with this key, the lock would be opened so that this and the viral genome enters into the body. I mean, the cell body. I mean, the cell cytoplasm, and then it hijacks the machinery of the virus. Sorry, hijack the machinery of the human host cell, right? So this is the entry point. Just like how a burglary, it means that when thief avoids for the situation, right? When he enters into the house, he takes over, he hijacks. The same way, this new coronavirus also has this crown proteins or spike proteins, which acts like a key and which fits with with the host receptors, acts like a lock, so that it would fuse with the plasma membrane of the cell, because we are animal, we ourselves are animals. So in the sense we have no plant, we have no cell walls, etc. So we have only one membrane, plasma membrane. So this virus fuses with this plasma membrane, and the genetic material of the coronavirus would be coming into the cell cytoplasm. So this is method of entry, actually, viral entry. So target of this new coronavirus, right? What is the target? The target is actually this throat cells or lung cells. So they go and find their locks and try to open the keys, right, with the help of this spike proteins. And you would have seen yesterday's news that DRDO, right? So this DRDO is DRDO with the in collaboration with Dr. Reddy's. They have developed a certain new drug which actually reduces the oxygen consumption, oxygen levels. Right now, what happens in coronavirus infected patients is that the oxygen saturation levels are very high. It means they require more number of oxygen molecules. So it reduces that, and they are using an analog of glucose molecule. So why glucose is that when they have to replicate, they use the sugar molecules, ribose sugars, nucleotides. So instead of that, they will be using these and they will stop the replication. So it will decrease. So this new drug go interacts with the already infected cells and they slow down the replication. So obviously it's controlling actually, right? So this is yesterday's news actually, the current news. So the drug that was released by DRDO, deoxy deoxyglucose. Yes. 2DG, 2-deoxy, 2DG. This is a drug name actually. 2DG anti-COVID drug launched by DRDO collaboration with Dr. Reddy's, right? But these are all, it comes in picture after a few days, but still this is a mechanism actually. So let's see what else we can see. Why I'm telling here about the antiviral drugs is when you know what is a point of entry, we can actually stop them. For example, imagine that the cell is like a home. Okay, let me show that. Guys, are you able to follow?
Yes, sir.
Like, imagine your cell. Okay, we know that. So cell as a house. Okay, let's assume this is fencing, etc. So imagine cell as a house, okay, and virus as a thief. So thief awaits for an opportunity. Okay. So thief target would be always immunity, right? Means if it can lower the gates or the security of the cell, obviously is this house as a person immunity, for example, right? Like that, sometimes there is a possibility that the key, for example, some security person or some other there is some there is a way that this virus is trying to enter into the house, right? And this coronavirus has this spike proteins which is a key, and there are certain receptors which if it is attached, then obviously infection would occur. So the F2 would occur. It means that when we know where exactly virus is trying to enter, we can stop that. That's what antivirals does. So antivirals, what we prepare, can be stopped at certain stages. It's not specifically it goes and kills only the virus, but that also is there. The antibodies which are released by vaccination. What happens in vaccination, we'll deal in last video. Vaccination results in production of antibodies against the virus. So they actually neutralize this virus without causing any infection. But this is naturally, the antibodies are produced, right? So this is one way. And other way is antiviral drugs. What they do is we have to identify that what is the point of entry. If you know the entry, we can stop it. Likewise, viral life cycle, even HIV also. Right now, everyone is talking about coronavirus, but the more deadliest virus would be actually HIV. So, I mean, the transmission is only through body fluids. Okay. If in sex and other aspects, we'll deal that in 12th standard. But the viral life cycle have a similar mode of infection. That a cell, human cell is there, and it has only plasma membrane. Plasma membrane contains lipids and other proteins like this receptors or some other channels, and nucleus, double membrane nucleus, right? So the virus which comes, so the virus which is coming, right? It tries to attach to this plasma membrane. Once it attaches, the genome of this, the genetic material of this virus would be going inside. And the machinery to replicate this will be provided by these cells. I mean, provided by the virus itself. So what exactly the antiviral drugs does is they try to stop this viral attachment at this. Okay, if they are attached, at least they will try to stop, you know, uh, this RNA or DNA, the genetic material moving inside the host nucleus. If, for example, if they are already integrated, it means they are already moved into the host cell. Then what happens? This viral DNA will be part of our DNA. That's what our cell cannot differentiate between viral gene and our gene, because each of everything is made up of the same raw materials. It cannot find out. So then what happens? Protein synthesis will start. So here we can stop. Protein synthesis we can stop. So any one of the steps, if we can stop, the replication of this virus will be slowed down. So when the number of viral particles are coming down, it means we can manage the infection, and obviously we can treat that person. That's what the 2DG, drug released by Dr. Reddy's and DRDO, India. So find out. So if you see this DRDO, Defense Research and Development Organization. So like that we have CCMB. So these guys are all national institutions, right? Where you can do the research. Even we try to do like that. We have HCU, University of Hyderabad, where I have done my research. Likewise, we can collaborate with these guys and we can, for example, bio toilets. So these are all the guys, ICMR. These are all national institutions where they develop these anti-drug, antiviral drugs and other scientific advancements. Okay. So like this, these drugs are available. So that's why I'm trying to tell, when you really understand how a thief can enter the house, so that we can provide with the entire fencing mechanism. Okay, backside we have to make a fencing, and we have to have these many doors and lock mechanism. So likewise, even we need to develop an antiviral drug, we need to understand its mode of infection. Understood? So let's move forward. So I told now. So this is a key and lock. So there is a receptor present on our cells, throat cells and lung cells, so that this yellow color lock and this blue color spike proteins will be locked with along with this key and lock, and then they try to enter into the cell like this. So actually, HIV virus has a life cycle where only the genetic material enters into the cytoplasm. But in a typical coronavirus, flu viruses, along with the plasma membrane, they will be entering into the cytoplasm. So here what happening is, even the nucleus doesn't, even the coronavirus, it no need to go into the nucleus as well. But with the presence of these ribosomes within the cell, machinery, so they start producing replicating. So ribosomes, where it replicates or makes more number of copies and proteins, so that directly they are producing the viral proteins and the virus itself. See, this is how a new viral virus is producing. So we have already seen the pneumonia, and there is a breathing difficulties and oxygen saturation, right? So how is that happening when this coronavirus is infecting throat and lung cells? So pneumonia development, and what exactly happens? Pneumonia, we already seen how pneumonia would develop, and the inflammation and the fluid accumulation, and the air sacs would be, inflammation would take place, right? And you can see that the oxygen concentration would be lowered. So normally we cough off whatever the particles which are there, irritants and etc., dust as well. Otherwise, our immune cells like macrophages would attack the unnecessary foreign particles. But when the infection progresses, So the same way how in coronavirus patients, what's happening is when the immunity is weakened, obviously the bronchiole, this air sacs at the bronchioles getting inflamed. Inflammation is seen because they are thoroughly attacking this immune cells, etc. So they start swelling and then finally fluid accumulation. So if a air balloon has 3/4 of the water, the air present inside is only 1/4, right? The same way, air sacs, the volume is actually almost 3/4 volume is filled by this fluid. So obviously, what we are getting is 1/4 of oxygen. It means our oxygen demand is increasing and the oxygen availability is decreasing because of the fluid accumulation. So if it goes on and goes on, if the infection progresses further stages, even the difficulty in breathing would be increasing. Then we have to put the patient on ventilator, which is a mechanical breathing system, provides the oxygen. So this is vaccine development. The last part what we are going to discuss is vaccine development, drugs and vaccines. So how exactly vaccine works? So let's see that. So far we try to understand that we try to protect, fence our house with immunity, etc. Okay, so vaccination is something like a drill, mock mock drill. For example, you bought a house, okay, and it is fully automated. Okay, fully automated. Everything would be under surveillance, CC cameras, etc., etc., etc. Security systems. So as a new to this person, right? You are asking for an demo, okay? Demo or mock drill. It means a thief would try to enter into the house, and we will check all the security systems, right? Likewise, a vaccine is a virus, not active, but it is partially virus and it can trigger the immune response, just like how an actor goes into the house as a thief, it triggers the alarms and etc etc, like that. During the mock drill, the same way virus, which can be an half of the virus or part of the virus, or some molecules of the virus can be prepared as, or sometimes the virus can be inactive. For example, in the case of polio virus, polio vaccine, so it is inactivated. Inactivated as in the polio virus, they take polio virus and they treat with certain chemicals to make it inactive. It will not replicate. And they give this as a vaccine. So that what happens, it triggers the immune response as if really it's a virus, and then it starts producing antibodies against it. And once when the real thief enters, obviously we will be stopping this guy, right? We will be checking. Before we already checked it, right? So memory. So our body will have a memory B cells, immune cells. This topics immunity would come in your 12th standard, but I'm giving basics here. So B cells produces these antibodies that memory. This just like how police station will have thief profiles, photo and details. Likewise, the B cells will have all the profiles of the virus, bacteria, fungus, other infectious agents, and they once this vaccine was given against this virus, it tried to recognize this virus, and later it would be the response of immunity would be so high, aggravated response would be seen. And finally, what happens when a person really infected with the coronavirus, it would be stopping the coronavirus because it already had antibodies against this coronavirus or polio, whatever the virus is. But right now we are talking about coronavirus. So it can stop. So all the immune cells would attack this coronavirus and stops its infection. That's how vaccines work. Okay. So antivirals would maintain, immediately it kills, but in a lifelong immunity is provided by this vaccines. Did you understand the analogy? Are you able to follow? Guys, did you understand the analogy? Yes or no? We have 10 minutes time.
Sir,
Okay, fine. So let's move forward. So that's how vaccine works. For example, when the antibodies are attached to these keys, okay? So there are keys on the coronavirus protein code, right? So these keys are masked by antibodies so that it cannot really open the lock of our host cells. So obviously the goal is achieved. So that the viral particles which are floating in the blood, which are there, they will be attacked by this microorganisms. Attacked by this immune cells, macrophages, etc. They will eliminate them. So in later stages, even if a real virus comes, it will be attacking those virus in a more aggravated manner, and they will make this into pieces because the virus is made up of different molecules. So obviously it would be chopped off in the macrophages, digest completely. So here the vaccine development has several stages. For example, we have to see that the vaccine we develop has to work on cell level, and then some lab animals, lower organisms, mammals, and then we have to go for human trials. And then we have to go for the pharmaceutical, I mean, placebo. There are several stages, but the record-breaking vaccine preparation in previous Ebola virus vaccine, where it took almost 5 years, which is a short span. And Edward Jenner, our father of vaccines, I have to place a photo of him in my house actually, because as a microbiologist, I treat them as actual saviors of life. Louis Pasteur, Edward Jenner, Anthony von Leeuwenhoek. If these three guys are not there, obviously the population of entire world would be just 1 billion, something like that. with still more diseases. But at the advent of invention of microscope, Antonio and Leeuwenhoek to Louis Pasteur's discoveries, biogenetic theory, and as well as the other pasteurization techniques, and even Edward Jenner's vaccine, cowpox, and all this elimination has done successfully right from once the science has developed. And now the COVID-19 vaccine is prepared almost just within a year time, 12 months time, is a record breaking for entire history of the medicine vaccines.